220 million years ago, something crawled across the mudflats of what is now southern China that would outlast nearly every complex organism on the planet. It had a partial shell covering its belly, teeth lining both jaws, and a body built for shallow coastal waters. Paleontologist Li Chun at the Chinese Academy of Sciences identified it from fossils recovered in Guizhou province and published the findings in Nature in 2008. He named it Odontochelys semi-testacea, the toothed turtle with half a shell. It was the oldest turtle ancestor ever found with clear evidence of shell development, and it rewrote everything biologists thought they knew about when and how the most iconic structure in the reptile kingdom first appeared. That animal, or something extremely close to it, survived what came next. It survived the end-Triassic extinction 201 million years ago, when volcanic eruptions across the central Atlantic magmatic province released enough carbon dioxide to raise global temperatures by roughly 5 degrees Celsius and wipe out approximately 76% of all species on Earth. It survived the Jurassic Ocean anoxic events that suffocated marine ecosystems across entire ocean basins, killing everything that depended on dissolved oxygen in the water column. It survived the end Cretaceous extinction 66 million years ago, when an asteroid roughly the width of Manhattan slammed into the shallow seas off what is now the Yucatan Peninsula and killed every non-avian dinosaur that had ever lived. That was 66 million years ago. Turtles walked out of that catastrophe and into the Cenozoic era without missing a geological beat. That was 66 million years ago. Today, a plastic grocery bag manufactured in a factory outside Shenzhen, stamped with a store logo, used for 11 minutes to carry a carton of eggs from a checkout counter to a kitchen table, and then tossed into a drainage ditch that feeds a river that empties into the South China Sea. That bag is killing them. Not metaphorically, not in some slow, abstract, hand-wringing statistical way. A sea turtle encounters that bag drifting through open ocean water, mistakes it for the one food source its body has spent 100 million years evolving to catch, swallows it whole, and dies. The bag blocks the intestine. Or it fills the stomach with indigestible material until the turtle stops eating, because its body registers fullness that never goes away. Or it lacerates the gut lining and triggers a slow internal infection that takes weeks to finish the job. And this is happening to an animal that survived 5 mass extinctions. An animal whose body plan has remained fundamentally intact for longer than the Rocky Mountains have existed. How is that possible? The answer is not simple. And it starts much further back than most people realize. The very adaptation that made turtles nearly indestructible for 220 million years, the same biological engineering that carried them through asteroid winters and volcanic apocalypses and ice ages that buried continents under kilometers of glacial ice, is the exact mechanism that now delivers plastic into their stomachs with surgical efficiency. That irony only becomes visible when you understand what their bodies were actually built to do and how long it took to build them. In 2015, paleontologist Rainer Schoch at the Stuttgart State Museum of Natural History in Germany published a study in Nature describing a fossil he had excavated from 240 million-year-old limestone deposits near the town of Velberg in Baden-Württemberg. He named it Papachelys rosinae, grandfather turtle. The animal was small, roughly 20 centimeters from snout to tail, about the length of a standard school ruler. It had no shell. What it did have were broadened ribs, thickened and beginning to fuse in a pattern that no other reptile of that geological period exhibited. Schoch interpreted these ribs as the earliest visible stage of shell development, a skeletal framework that was slowly widening and flattening in a trajectory that, given another 30 million years of incremental refinement, would eventually close into the complete bony casing you see on every turtle alive today. But the full timeline of that transformation stretches even further back. In 2013, Tyler Lyson at the Denver Museum of Nature and Science re-examined fossils of a creature called Eunotosaurus africanus, a 260 million-year-old reptile from South Africa's Karoo Basin. Lyson published his analysis in Current Biology, and the findings were striking. Eunotosaurus had 9 pairs of dramatically broadened ribs, reduced intercostal muscles between those ribs, and a body profile that was already trending unmistakably toward the turtle form. Not a turtle yet. but something that was clearly, measurably heading in that direction. 260 million years before the present. The ribs were widening. The spine was stiffening. The body was compressing laterally. Evolution was building a tank, and it was taking its time. From Unitosaurus at 260 million years, to Papaschalus at 240 million, to Odontochelys at 220 million, to Progonocales quenstediae at roughly 210 million years ago, the first turtle known to have a fully enclosed dorsal and ventral shell, the evolutionary progression is preserved in the fossil record like a sequence of engineering prototypes. Each iteration added reinforcement. Each generation thickened the armor, until what finally emerged was one of the most successful body plans in the entire 500 million year history of vertebrate life on Earth. That shell is not a house the turtle carries. It is the turtle. The shell is fused to the spine and the ribcage at the structural level. A turtle cannot crawl out of its shell any more than you could crawl out of your own ribcage. The ribs do not sit inside the body cavity the way yours do. They are the outer wall of the body cavity. And the shoulder blades, which in every other vertebrate on the planet sit outside the ribcage, are located inside the shell in turtles. This anatomical reversal, the shoulder girdle tucked beneath the ribs, exists nowhere else in the animal kingdom. It is a completely unique skeletal arrangement that took roughly 50 million years of gradual restructuring to achieve. If you want the complete breakdown of how evolution, extinction, and deep-time survival all connect, including the full research framework behind the kind of science covered in this video, check out the ebook linked in the description below. It goes deeper than any single video can, and it covers the system behind these stories, not just the stories themselves. Now, the shell worked. For 220 million years it worked. So when the single worst day in the last 250 million years of Earth's history arrived, the day that ended the age of the dinosaurs, what happened to the turtles? 66 million years ago, an asteroid roughly 10 to 15 kilometers in diameter struck the shallow waters of what is now the Gulf of Mexico at an estimated velocity of 20 kilometers per second. To put the energy release in perspective, the combined nuclear arsenals of every nation on Earth, detonated simultaneously, would produce roughly 1/15 of the energy released by the Chicxulub impact. The blast wave flattened forests across North America. Superheated debris launched into the upper atmosphere rained back down as molten glass beads, igniting firestorms across 3 continents simultaneously. Sulfur and soot blocked sunlight for months. Some models suggest photosynthesis effectively ceased for over a year across large portions of the planet. Global temperatures collapsed. The food web disintegrated from the base upward, starting with photosynthetic organisms and cascading through every animal that depended on them. 76% of all species on Earth went extinct. The non-avian dinosaurs that had dominated terrestrial ecosystems for 165 million years, longer than the entire elapsed time between their extinction and the present day, were erased. The mosasaurs that had been the apex predators of the world's oceans vanished entirely. The pterosaurs that had ruled the skies for 150 million years disappeared without a trace. The turtles survived. Not all of them. The fossil record shows clearly that several turtle lineages went extinct at the Cretaceous-Paleogene boundary, particularly larger-bodied species and those with specialized diets tied to stable terrestrial food webs. But the order Testines as a whole, the clade that includes every turtle, tortoise, and terrapin alive today, persisted through the catastrophe. Freshwater turtles and marine turtles in particular made it through. In 2019, Tyler Lyson published a landmark study in the Proceedings of the Royal Society B, analyzing turtle fossils from the Denver Basin in Colorado, one of the most complete and continuous post-extinction fossil assemblages anywhere in the world. The Denver Basin preserves rock layers spanning the period immediately before, during, and after the Chicxulub impact with remarkable resolution. What Lyson's team found was that freshwater turtles not only survived the extinction event, but recovered their pre-impact diversity faster than almost any other vertebrate group in the record. Within roughly 100,000 years, geologically instantaneous by any measure, turtle diversity in the Denver Basin was already rebounding toward pre-extinction levels. faster than mammals, faster than birds, faster than crocodilians. The reasons trace directly back to the biological toolkits that 150 million years of prior evolution had already assembled. Turtles can enter a state of torpor. In cold conditions, freshwater turtles can slow their metabolic rate to near zero, surviving for months without food, sometimes buried in mud at the bottom of ponds and rivers, with almost no measurable oxygen consumption. When the impact winter blocked incoming sunlight and crashed global temperatures, animals that depended on active foraging burned through their energy reserves and starved. Turtles that could simply shut down and wait underground or underwater had a different equation. Turtles are dietary generalists. They eat plants, insects, small fish, carrion, algae, fungi, whatever the environment provides. When the food web collapsed and entire categories of prey disappeared, the specialist feeders, the animals that could only eat one type of food, disappeared with their food source. Turtles ate whatever was still available. A turtle that can survive on pond algae and decomposing plant matter does not need a functioning vertebrate food web to persist. Freshwater systems partially buffered the worst chemical effects of the impact. The acid rain that sterilized large areas of the ocean surface and devastated terrestrial soils was diluted in freshwater rivers and lakes. The pH swings that proved lethal to marine organisms with calcium carbonate shells were less extreme in freshwater environments. Turtles living in rivers and ponds were shielded from the worst of the atmospheric chemistry. And they had the shell, 250 million years of structural engineering, fused to the skeleton, protecting them from the debris falls, the temperature swings, the desperate predation attempts by starving survivors competing for the same dwindling resources. This is where the story takes a turn nobody expected. Every single one of those survival advantages works against threats that alter the external environment. Temperature collapses, atmospheric poisoning, food web disruption, predator pressure, chemical shifts in water. The turtle body plan is a masterpiece of environmental resilience. Evolution spent 220 million years building an organism that could survive the world changing around it in extreme and catastrophic ways. What evolution did not do, what it could not have done, was prepare turtles to survive a world that manufactures objects visually identical to their primary food source, and then distributes those objects across every ocean on the planet in quantities measured in the trillions. Sea turtles, particularly leatherback sea turtles, evolved to eat jellyfish. This was not a casual dietary preference or a seasonal supplement. Jellyfish feeding is a deep specialization that has defined leatherback biology for tens of millions of years and shaped every aspect of their physiology. Leatherbacks are the largest living turtles on Earth. Adults regularly reach 550 to 700 kilograms, with the largest recorded individual weighing over 900 kilograms, roughly the mass of a small car. They are also the deepest diving reptiles known to science, capable of reaching depths exceeding 200 meters below the surface, deeper than most military submarines are rated to operate. And they accomplish all of that on a diet composed almost entirely of jellyfish, an animal that is approximately 95% water by weight and contains so little caloric energy that a leatherback must consume an extraordinary volume of them to maintain its metabolic needs. A 2009 study by Mike James at Dalhousie University in Nova Scotia estimated that a foraging leatherback may need to consume up to 73% of its own body weight in jellyfish every single day just to sustain its energy budget. For a 600-kilogram turtle, that is roughly 440 kilograms of jellyfish daily, more than the weight of 4 adult humans eaten in a single 24-hour feeding cycle. I mean, think about that for a second. An animal that weighs as much as a grand piano eating nearly its own body weight in translucent, near-zero-calorie prey every day. This is only possible because of a specific anatomical adaptation that evolved over millions of years to solve a very particular engineering problem. Jellyfish are slippery. They have no bones, no rigid structures, no textured surfaces. A smooth, gelatinous blob, the consistency of wet plastic wrap sliding through an esophagus designed for other food types, would simply slide right back out. The leatherback solved this problem with papillae, hundreds of backward-pointing keratinous spines lining the interior of the esophagus. These structures, some measuring several centimeters in length, create a one-way system. a jellyfish enters the throat and physically cannot slide back out. The papillae grip the gelatinous body from every angle and force it downward toward the stomach. Without them, the leatherback's jellyfish diet would be mechanically impossible. The papillae are the engineering key that unlocks the entire strategy. This adaptation was, for 100 million years, one of the most elegant feeding solutions in the marine world. Jellyfish are among the oldest multicellular animals on Earth, with a fossil record stretching back over 500 million years. Their populations bloom reliably and massively in warm, nutrient-rich waters. They have no defensive spines, no hard shells, no venom potent enough to deter a large predator. For a marine reptile willing to eat in the enormous volumes necessary to compensate for the low caloric density, jellyfish were the perfect food source. Abundant, defenseless, predictable across geological time. Leatherbacks and their evolutionary predecessors followed jellyfish blooms across the planet's oceans for 100 million years, migrating thousands of kilometers between breeding grounds in the tropics and feeding grounds in temperate waters, tracing the seasonal pulses of their gelatinous prey with navigational precision that scientists still do not fully understand. The papillae gripped, the jellyfish went down, the turtle survived. For 100 million years, the system ran without a fault. And then, in 1965, a Swedish engineer named Sten Gustaf Thulin, working for a packaging company called Celloplast in the town of Norrköping, filed a patent for the modern t-shirt plastic bag. Thulin's design was simple, heat-sealed from a tube of polyethylene film, cheap to manufacture, lighter than paper, and waterproof. The handles were cut from the same piece of film, no stitching, no rivets, no separate components. Just a single sheet of polymer folded, sealed, and cut into a shape that could carry groceries. Within 15 years, plastic bags had replaced paper bags in grocery stores across Europe, the United States, and East Asia. By 2023, the United Nations Environment Program estimated that humans were producing approximately 5 trillion plastic bags per year worldwide. 5 trillion. That number is difficult to grasp at any intuitive scale, so consider it this way. 5 trillion plastic bags per year means approximately 160,000 bags manufactured every second. If you laid a single year's global production of plastic bags end to end, the line would wrap around the circumference of the Earth more than 40,000 times. That is enough plastic bags to stretch from Earth to the Sun and back with material to spare. The average use time of a single plastic grocery bag is 12 minutes. After those 12 minutes, the bag enters the waste stream. And in the dozens of countries that lack comprehensive waste management infrastructure, the waste stream is not a landfill. It is a river. And the river empties into the ocean. Pay attention to this next detail because it connects every piece of this story. A translucent plastic bag drifting in open ocean water is, to the visual system of a sea turtle, indistinguishable from a jellyfish. The silhouette is similar. The way it catches ambient light and filters it through the semi-transparent material is similar. The way it drifts with the current, pulsing and folding with the movement of the water, mimics the rhythmic contraction of a living jellyfish with disturbing accuracy. And the visual recognition system of a sea turtle, refined across 100 million years to identify jellyfish from a distance in murky, particulate-heavy ocean water, was never calibrated to distinguish between a living cnidarian and a sheet of petroleum-based polymer film. The turtle sees the bag, the ancient recognition pathway fires, the approach behavior initiates, the mouth opens. The papillae do exactly what 100 million years of evolution designed them to do. They grip the object and force it downward, past the point of no return, into the stomach. And this is the detail that changes the math entirely. The backward-pointing spines that were the turtle's greatest evolutionary asset become, in this exact moment, the instrument of its death. The papillae that prevent a jellyfish from sliding back up the esophagus also prevent a plastic bag from being regurgitated. The turtle cannot spit it out. The bag is mechanically locked inside the digestive tract by the very structures that evolution spent 100 million years perfecting for a completely different purpose. The system works exactly as designed, and the result is fatal. In 2018, Chris Wilcox at Australia's Commonwealth Scientific and Industrial Research Organisation, CSIRO, published a study in the journal Scientific Reports that quantified the relationship between plastic ingestion and sea turtle mortality for the first time with statistical precision. Wilcox and colleagues analyzed data from 246 turtle necropsies conducted across Australian coastal waters, examining the gut contents of animals that had died and washed ashore. The findings were precise and devastating. A sea turtle that had ingested a single piece of plastic had a 22% probability of death from that ingestion alone, not from a stomach full of plastic, from one piece. A turtle that had consumed 14 pieces, roughly the amount of material in a single shredded grocery bag, had a 50% probability of death. The mortality curve was not gradual, it was steep, and it started from the very first item. And no, that is not a typo. One piece of plastic. 22% chance of dying. Kamar Schuyler at the University of Queensland had published complementary research in 2015 in the journal Global Change Biology, analyzing over 300 sea turtle necropsies from Australian and North American waters. Schuyler's team found that younger turtles were disproportionately affected by plastic ingestion, likely because juvenile sea turtles feed closer to the surface in oceanic convergence zones, the exact areas where floating debris accumulates in the highest concentrations. In some sampled populations, over half of all necropsied individuals had plastic fragments inside their digestive tracts. The problem is not localized. It is not confined to one coastline or one ocean basin. A 2015 analysis published in the journal Science by Jenna Jambeck at the University of Georgia estimated that approximately 8 million metric tons of plastic enter the world's oceans every year. 8 million metric tons. That is the equivalent of backing up a full garbage truck to the shoreline and dumping its entire load into the surf every single minute of every day for 365 days straight. The material does not biodegrade. It photodegrades, meaning ultraviolet radiation from sunlight breaks it into smaller and smaller fragments over time. But the polymer chains themselves persist for centuries. A plastic bag manufactured in 1985 is still in the ocean right now. It has fragmented into thousands of particles, each one small enough to be swallowed by a fish, a seabird, a filter-feeding organism, or a sea turtle hatchling. In 1997, Charles Moore, a sailor and ocean researcher who would go on to found the Algalita Marine Research Foundation, sailed through an area of the North Pacific between Hawaii and California and encountered a vast, diffuse field of floating plastic debris stretching to the horizon in every direction. He described it publicly as a garbage patch, and subsequent surveys by the Ocean Cleanup Foundation and other research groups confirmed its scale. The Great Pacific Garbage Patch covers an area roughly 3 times the size of France. It is not a solid floating island that you could walk across. It is a 3-dimensional cloud of suspended plastic particles and larger debris items, stretching across the surface and descending through the water column to depths where light barely penetrates, creating a toxic corridor that marine animals cannot detect, cannot navigate around, and cannot avoid. Leatherback sea turtles migrate directly through these accumulation zones. Their annual migration routes, among the longest of any reptile, spanning thousands of kilometers from tropical nesting beaches to temperate and subarctic feeding grounds, overlap with every major oceanic garbage patch on the planet. The North Pacific Garbage Patch, the North Atlantic Garbage Patch, the Indian Ocean Garbage Patch. The turtles cross them because the jellyfish are on the other side. They have been crossing these same ocean corridors since before the Himalayas existed. The plastic arrived in those corridors 50 years ago. The turtles have no evolutionary basis for recognizing it as anything other than food. If you have made it this far, and this kind of deep science narrative is the content you look for on this platform, subscribe to the channel right now. I cover these intersection points between deep evolutionary history and the modern world every week, and the next video in this series makes this one look like a warm-up. While the plastic ingestion crisis alone would be enough to threaten populations that have been stable for geological epochs, it is not the only modern hazard exploiting ancient turtle biology. Something else is happening to sea turtles right now that operates on a completely different axis of vulnerability, and it is connected to a reproductive mechanism so old that it predates the appearance of flowers on Earth. Sea turtles do not have sex chromosomes. This is a critical distinction. In mammals, biological sex is determined at the moment of conception by whether the fertilizing sperm carries an X or a Y chromosome. The environment after conception plays no role. In sea turtles, the opposite is true. Sex is determined not by genetics but by the temperature of the sand surrounding the eggs, during a critical window of embryonic development, typically the middle third of the incubation period. This system is called temperature-dependent sex determination, and it has been the standard reproductive mode for turtles since before the first tyrannosaur walked the earth. In most sea turtle species, the pivotal temperature sits near 29 degrees Celsius, roughly 84 degrees Fahrenheit. Eggs incubated in sand below that temperature produce predominantly male hatchlings. Eggs incubated above it produce predominantly females. The transition zone is narrow, sometimes just 1 to 2 degrees Celsius wide. A nesting beach with average sand temperatures of 28 degrees produces a clutch-skewed male. The same beach at 30 degrees produces a clutch that is overwhelmingly female. For over 200 million years, this system was self-correcting. Sand temperatures on nesting beaches fluctuated within ranges that produced a roughly balanced sex ratio across populations over time. Some clutches in warmer microclimates skewed female, others in shadier or cooler spots skewed male. Across an entire nesting season, across decades and centuries, the ratios averaged out. The system worked because the climate stayed within the envelope that turtles had adapted to across geological time. In 2018, marine biologist Michael Jensen at the National Oceanic and Atmospheric Administration, working with Cameron Allen at NOAA's Southwest Fisheries Science Center and colleagues, published a study in the journal Current Biology that quantified what warming temperatures were doing to this system on the Great Barrier Reef. Jensen's team used a combination of endocrine analysis and genetic testing to determine the sex of green sea turtles from two distinct breeding populations, one from the cooler southern Great Barrier Reef and one from the warmer northern section. The southern population was approximately 65 to 69% female, skewed but within a range where enough males existed to sustain reproduction. The northern population was 99.1% female. And among the juvenile turtles sampled from the northern rookeries, the ones most recently hatched, 99.8% were female. Functionally, every single hatchling emerging from the northern Great Barrier Reef nesting beaches was female. Jensen's team correlated the sex ratio data with sea surface temperature records spanning several decades, and concluded that the northern rookeries had been producing predominantly female hatchlings for more than 20 consecutive years. The arithmetic is merciless. A breeding population can sustain itself with a female-skewed sex ratio as long as enough males survive to fertilize the eggs. Green sea turtles can live 80 years or more. The males currently fertilizing eggs at the northern Great Barrier Reef were born decades ago, before sand temperatures consistently exceeded the pivotal threshold. Those males are aging, and the pipeline of replacement males from the northern beaches has been functionally shut off for 20 years. When those last males die, there will be no younger males to take their place. The population does not crash immediately. It crashes in 30 to 50 years when the final generation of males born before the temperature shift reaches the end of its natural lifespan. The extinction event is already programmed. It is just running on a delayed timer. This is happening on nesting beaches across the tropics. The northern Great Barrier Reef is not unique. Every nesting beach on Earth that experiences sustained warming past the pivotal temperature range undergoes the same shift. The Caribbean, the coast of West Africa, the beaches of Southeast Asia. The sex determination system that balanced turtle populations across 200 million years of climate variation is being overwhelmed by a rate of temperature change that exceeds anything in the recent geological record. And evolution cannot fix it fast enough. Temperature-dependent sex determination is encoded deep in the turtle genome. It is not a learned behavior. It is not a flexible physiological response. It is a fixed developmental pathway. For turtles to evolve chromosomal sex determination, the kind that mammals and birds use, they would need tens of thousands of generations under sustained selective pressure. At roughly 25 to 30 years per generation for most sea turtle species, that is hundreds of thousands of years, minimum. Global average temperatures have risen approximately 1.1 degrees Celsius above pre-industrial levels in the last 150 years. The Intergovernmental Panel on Climate Change projects an additional 0.5 to 3 degrees of warming by the end of this century depending on emissions pathways. The climate is changing on a scale of decades. the evolutionary response requires a scale of millennia. The gap between those two timelines is not a nuance. It is the gap between survival and extinction. And the mismatches do not stop at sand temperature. Sea turtle hatchlings emerge from their nests at night. This is not random timing. Nighttime emergence is an evolved anti-predation strategy. Hatchlings that emerge during daylight are immediately visible to gulls, frigate birds, crabs, and every other predator patrolling a tropical beach. Emerging at night reduces that risk dramatically. But the hatchlings still need to find the ocean. They are born in a hole in the sand, typically 30 to 60 centimeters deep, with no view of the horizon. When they claw their way to the surface, they need an orientation cue. And the cue that evolution provided, calibrated over millions of years on nesting beaches, where the only light sources were celestial, is simple. Move toward the brightest part of the horizon. For millions of years, the brightest part of the horizon on a nesting beach at night was the open ocean. Moonlight and starlight reflecting off the water surface created a diffuse glow that was consistently brighter than the dark silhouette of the forested or vegetated landward horizon. The hatchlings oriented toward that glow, scrambled down the slope of the beach, and reached the water. The system was elegant, reliable, and required no learning, no parental guidance, no trial and error. In 2016, a comprehensive review by conservation biologists at Florida Atlantic University documented the consequences of introducing artificial light to nesting beaches. On developed coastlines in Florida, the Caribbean, Mexico, and coastal Australia, the light output from beachfront hotels, resort complexes, restaurants, parking garages, highway overpasses, and residential buildings now overwhelms the natural brightness differential between the ocean horizon and the land horizon. The artificial sources are brighter. The hatchlings orient toward them. And they walk in the wrong direction. They walk away from the ocean and toward the coastal road. They walk into parking lots and become trapped in storm drains. They walk toward swimming pool lights and fall in. They walk in circles beneath street lamps until they collapse from dehydration and exhaustion, sometimes dying just meters from the waterline they were trying to reach. On some heavily developed beaches in Florida during peak tourist season, disorientation events affect the majority of emerging hatchlings in a given nest. If you are wondering how that is even legal, so is everyone working in turtle conservation along the Florida coast. The turtles are executing the program flawlessly. Move toward the brightest light. That is what the software says. That is what it has said for 100 million years. The software was never updated for LED floodlights because no evolutionary mechanism exists to issue that update on a timescale of decades. And even the turtles that avoid the plastic, survive the skewed sex ratios, and successfully navigate past the artificial lights still face one more gauntlet that their evolutionary history left them completely unprepared for: commercial fishing gear. Long-line fishing vessels deploy monofilament lines that stretch for over 100 kilometers across open water, baited with thousands of hooks. Bottom trawl nets scrape the ocean floor across areas where turtles feed and rest on the substrate. Driftgill nets hang like invisible curtains through the water column, indiscriminately catching everything that swims into them. A 2010 analysis by Brian Wallace at Duke University estimated that longline fisheries alone hooked approximately 85,000 sea turtles per year in observed fisheries globally. That figure only accounts for fisheries carrying onboard observers who documented and reported the catches. The actual number, including unreported bycatch across unmonitored fleets in the Indian Ocean, the South China Sea, and the Eastern Pacific, is almost certainly significantly higher. A sea turtle that has survived the plastic, dodged the temperature lottery, navigated past the artificial lights on its natal beach, crossed an ocean full of microplastic contamination, and successfully reached its feeding grounds, can still drown in a gill net in 3 minutes. Here is the detail that keeps researchers awake at night. The mismatch between evolutionary time and human industrial time is not a subtle academic distinction. It is the central crisis of modern conservation biology, and sea turtles are its clearest case study. Consider the timeline. Turtles in their broadest form have existed for roughly 260 million years, since Unitosaurus was crawling through the Karoo Basin. Plastic in any form has existed for 119 years, since Leo Baekeland synthesized the first fully synthetic polymer in his laboratory in Yonkers, New York in 1907. The polyethylene plastic bag has existed for 61 years. The high-output LED streetlight that disorients hatchlings has been widely deployed for approximately 15 years. The pace of global warming that is overriding temperature-dependent sex determination has accelerated meaningfully only in the last 50 years. Against 260 million years of evolutionary history, these threats represent less than 0.00005% of the time that turtles have been on Earth. It is the equivalent of an organism that successfully completed 260 consecutive marathons being killed by a crack in the pavement during the final 5 centimeters of the last race. The engineering held across 260 million years of the most extreme environmental stress the planet could deliver. It is failing now because the nature of the threat has changed in a way that 260 million years of experience provided no preparation for. Evolution does not work on demand. It does not identify a problem and generate a solution. It works through random genetic variation filtered by differential survival. The mutations that might allow a sea turtle to distinguish a plastic bag from a jellyfish, or to override the phototactic behavior encoded in its hatchling nervous system, or to develop chromosomal sex determination instead of temperature-dependent sex determination, would each require thousands of generations under sustained selective pressure. And even under ideal conditions, there is no guarantee the necessary mutations would ever appear. Genetic variation is random. Natural selection can only act on what variation provides. The turtles that ate plastic and died did not pass on any hypothetical plastic avoidance gene to their offspring. They simply died. Their genes exited the population. The hatchlings that walked toward the hotel lights instead of the ocean did not report back to the nest and warn their siblings. They dehydrated on the asphalt. Natural selection requires that the survivors be the ones carrying the advantageous variation. When the threat kills, essentially at random, when every turtle in a population encounters plastic that looks identical to jellyfish regardless of that individual's particular genetic makeup, selection has nothing differential to work with. There is no plastic-resistant turtle being favored. There is just death, distributed evenly across every genotype. There is a concept in conservation biology that captures this dynamic precisely. It is called an evolutionary trap. An evolutionary trap occurs when an organism's evolved behavior, a behavior that was highly adaptive in the environment where it originally evolved, becomes maladaptive, because the environment has changed faster than the organism can respond. The classic example is the moth that spirals into a flame. For millions of years, moths navigated at night by maintaining a constant angle to the moon, an effectively infinite light source. The system worked because no natural light source on Earth other than the moon was bright enough and persistent enough to hijack the behavior. Then humans invented fire, and the candle, and the porch light. And suddenly the navigation system that had worked for 190 million years of moth evolution was sending moths directly into lethal heat sources. The moth is not stupid. The moth is running ancient software in a modern environment. Sea turtles are caught in at least 3 evolutionary traps simultaneously. The jellyfish recognition system that worked without error for 100 million years is now a plastic ingestion pathway. The phototactic orientation behavior that guided hatchlings to the ocean for millions of years is now guiding them to parking lots. The temperature-dependent sex determination system that maintained balanced populations across geological epochs is now producing populations with virtually no males. Any one of these traps in isolation would be a serious conservation challenge. All 3 operating at the same time, on the same populations, alongside industrial fishing bycatch and habitat destruction on nesting beaches from coastal development, represents a convergence of threats that no amount of evolutionary history can compensate for. The turtles are not failing because they are poorly designed. They are failing because they are perfectly designed for a world that no longer exists. And this is where the story connects to something much larger than sea turtles. The same pattern of evolutionary mismatch is unfolding across thousands of species simultaneously. Albatrosses that evolved to feed their chicks squid and fish are scooping plastic debris from the ocean surface and delivering it to their nests. Laysan albatross chicks on Midway Atoll in the North Pacific are dying with stomachs packed with bottle caps, cigarette lighters, and plastic fragments, fed to them by parents whose food recognition systems cannot distinguish synthetic debris from prey at the water's surface. Insects that evolved to navigate by polarized moonlight are spiraling into LED streetlights by the billions, collapsing local pollinator populations in ways that ripple through entire plant communities. Coral reefs that persisted through 500 million years of ocean chemistry fluctuations, including the end Permian extinction that killed 96% of marine species, are bleaching and dying because the current rate of ocean acidification exceeds anything in the geological record by an order of magnitude. The common variable across all of these cases is speed. Every one of these species survived previous environmental disruptions, because those disruptions unfolded on timescales that allowed evolutionary responses to develop. The end Permian extinction, the worst biodiversity crisis in Earth's history, played out over tens of thousands of years. The KPG asteroid impact was geologically instantaneous, but the recovery period stretched across millions of years, providing ample time for surviving lineages to diversify and fill vacated ecological niches. Even the glacial cycles of the Pleistocene, which reshaped ecosystems across entire continents, operated on rhythms of 41,000 and 100,000 years, long enough for populations to shift ranges, adapt diets, and adjust reproductive strategies. The threats facing sea turtles emerged in less than a single century. Against 260 million years of evolutionary time, that is not a short interval. It is not even a rounding error. It is a single frame in a film that has been running since before the first dinosaur hatched from an egg. The turtle that crawls out of the Atlantic surf onto a nesting beach in Brevard County, Florida, on a warm night in August of this year is carrying 260 million years of tested, refined, proven survival engineering inside its body. Its shell was perfected across 50 million years of incremental skeletal restructuring. Its esophageal papillae were honed across 100 million years of jellyfish predation. Its hatchling orientation instinct was calibrated across eons of stable moonlight over dark, undeveloped ocean horizons. Its sex determination system was tuned to sand temperatures that stayed within a narrow, predictable range for longer than most mountain ranges have existed. None of that engineering included a subroutine for polyethylene film. None of it included a software patch for LED floodlights. None of it included a workaround for sand temperatures rising past the pivotal threshold of a reproductive system that has been running the same program since the Triassic. The operating system is 260 million years old, and the world it was written for has been rewritten in the last 60 years. The real question is not why turtles die from plastic bags. The real question is what it tells us when the planet's most durable survivors, animals that outlasted everything the Earth could throw at them for 250 million years, start dying from objects that have existed for less than a single human lifetime. Because the same evolutionary mismatch that is killing sea turtles is not unique to sea turtles. It is the operating principle behind the broader biodiversity crisis unfolding across every ecosystem on the planet right now. The mechanisms differ. The species differ. The specific mismatches differ. But the underlying equation is the same in every case. Ancient biology, shaped by millions of years of gradual environmental pressure, meeting modern disruption delivered at a speed that makes adaptive response impossible. The turtles have been demonstrating this equation for decades. Every carcass that washes ashore with a stomach full of plastic is a data point. Every nest of female-only hatchlings on a warming beach is a data point. Every hatchling circling under a streetlight until it collapses is a data point. The question is not whether the data is clear. It has been clear for years. The question is what we do with it. Because the machinery inside a sea turtle, the instincts, the drives, the hardwired responses that carried it through 260 million years of planetary catastrophe, is the same category of machinery that operates inside every complex organism on this planet, including the one watching this video. If you want to see what happens when this same evolutionary mismatch plays out on an even larger scale, and the species involved is not a turtle, click the video on screen now.